Good morning, and those who doesn't know me, I'm Karam Sadiq. I'm the Hackett Professor of Agriculture and the Director of UWA Institute of Agriculture here. Now, before we get started, it's customary for the University of Western Australia to say that we are situated on Noongar land, and the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, beliefs, and knowledge. On behalf of the University of Western Australia, I warmly welcome you to the special lecture by Dr. David Dent, which is jointly hosted by the UWA Institute of Agriculture and the UWA Institute of Advanced Studies. And we have a representative here, and they do a wonderful job, and we try to co-badge some of our key things together, and we will do that more. Now, our speaker, Dr. David Dent, is an agricultural scientist by training, which is quite important. You need to know what is agriculture before you talk about some of these things. An author of Inventor and Entrepreneur, an author of 6 books. His textbooks on insect pest management is now in its 3rd edition. And his latest work is on 2-volume monographs on the nature of scientific innovation, which is currently in press with Palgrave Macmillan. I look forward to get a copy, a free copy. I will buy, no problem. He's a visiting professor at the University of Surrey Business School for his development of an analytical technique for identifying market gaps for innovation. Again, an agricultural scientist turning into, School is development of an analytical technique for identifying market gaps for innovation, a former Royal Society entrepreneur in residence at the University of East Anglia, and a vice president of the UK's Parliamentary and Scientific Committee, as well as independent advisor to a UK government high-risk investment fund. Dr. Dent's inventions include a herbicide adjuvant, a sprayman's calculator, again very farmer-oriented, patents on nitrogen fixation, and the process to round up transactions at a point of sale, PIN terminals. This latter invention generates more than £1 million per year to disperse to UK charities through Penny's Foundation that he founded. So I was telling David this morning, a little bit of money to UWA will be good. Not for me, but we will work on that, because this is his first visit to Perth to his daughter, and we were saying that this is the best place in the world, and he agreed with me that. As a former managing director of CABI, again, those who all know CABI Bioscience, which is a very important international organization, do wonderful things, particularly in developing countries. He has also commercialized biopesticides for locust control in Africa, established a diagnostic company to dictate a fuel fungus for the airline industry. I don't know about that, we'll find out. And his latest company, Azotic Technologies Limited, commercialized an invention based on planned intracellular colonization by nitrogen-fixing bacteria that can substitute for synthetic nitrogen fertilizer in any crop. Today, he will be exploring the topic, fixed on nitrogen, challenging a paradigm to transform agriculture. Please welcome Dr. David Dent. Thank you, David. Yeah.
Thank you very much for that introduction, Professor Sadiq. It's a pleasure to be here with you all. It is my first time in Perth, and I must say the welcome here that I've had with people, the climate, I'm pretty much agreement that this is not a bad place in the world to be, is it? Goodness me. So today what I want to do for you really is to talk about the work of my Sustainable Nitrogen Foundation and the company Azotic Technologies Limited that I founded with a number of colleagues to look to see how we could take a nitrogen fixing concept in the laboratory all the way through to a field product that could be sold and is sold. But it might seem that that's a straightforward process, but it was far from that. And the reluctance and the resistance that we met along the way to making this happen is the story I'm going to tell you today. So, just something for you to mull over while I'm talking is, what if every green plant is capable of intracellular, meaning within cells, so the living cells, an intracellular symbiotic relationship with a nitrogen-fixing bacteria? What if every crop plant is capable of that, but it's also possible to replace, substitute for a significant amount of nitrogen that is usually provided by fertilizers while maintaining yields and perhaps even increasing yields? And what if modern crop breeding techniques have actually decreased our ability for these nitrogen-fixing bacteria to form these symbiotic relationships with our crop plants? We'll come back to these same points at the end of my presentation, and I'll be happy to discuss these and see what your views are and whether they may have changed as we go through my presentation. So there are some key terms here, and in the interest of always good communication, and so you can understand what some of the things I'm talking about when I don't make them clear each time, is we're talking about intracellular symbiosis. So this really is about living bacteria within cells doing their thing. And we're talking about biological nitrogen fixation, so the ability to take nitrogen from the air, fix it in a way that the plant can use, and release it into the plant. And the key point here is I'm talking largely about a nitrogen-fixing bacteria, Gluconosetobacter diazotrophicus. Now, I don't believe any of you want me saying Gluconosetobacter diazotrophicus all the way through the talk, so I'll be referring to it as GD. So please take note of that. And then I'll also talk about NFix, which is the seed inoculant technology. Not the final product, but the technology that went into all of our process of research and development. The technology leads to the product, which ends up with a product name that's completely different, but we had the working title of NFix. The other key point I want to make is about paradigms. And in science, we tend to, this is all the work of Thomas Kuhn back in the 60s, when he started talking about how science is conducted within paradigms, within a box. We define what our system looks like, we define what our methodologies, the concepts, the theories that are acceptable, and we work within that framework until something tells us, a lot of things tell us, that that framework no longer works. And we don't think of them on a day-to-day basis within science as often as we should. But we came up against this paradigm in the work that we're going to talk about today. So you should all know about paradigms with the work of Robin Warren and Barry Marshall. And believe me, it was a sheer delight. I hadn't realized you had the Barry Marshall library. And the photographs in there, I was in there this morning, absolutely brilliant, made my day. But this was paradigm shifting science. And one of the things that often doesn't get thought about is what the cost of that might be. And what's interesting about this, and I write extensively in my books on the nature of scientific innovation, which I will keep plugging, I talk about this paradigm of Barry Marshall because it's a great example of the issue, but it's also one of the most highly researched paradigm shifts. 25,000 papers have been published on this issue. Costs of paradigms. If we stick to one framework of thinking, don't think outside of the box, then scientists who challenge those paradigms, they face difficulties of getting work published, of securing grants to do further work, and reputational loss and even personal abuse. And I know this to be true. For society, it also means potentially a loss of time in getting an innovation out into the market. And this is something we face. But if you look at what Barry Marshall did, with the peptic ulcer disease, each year 0.5 million Americans are diagnosed with peptic ulcer disease. Every year that scientists did not come on board and agree with that paradigm shift, 0.5 million people in America still suffered from peptic ulcer disease. Look at that over 5 years. Look at that over 10 years. Unnecessary suffering. And the bit that gets me is those scientists opposing that change, no cost. No cost at all. So people can put barriers in the way and it costs them nothing. It's interesting that Niels Bohr, and the quote at the bottom, Niels Bohr basically said, really scientific change of paradigm size only ever comes about when scientists actually pass on and a new generation come along. It's very difficult to change paradigms. But that's what we're talking about. I want to just say something about, and this again refers back to my books, what is knowledge? We often don't necessarily think of that as scientists. So it's justified true belief is what we talk about. There's key elements here of being justified, of truth, where you're looking for reliable sources and verification and evidence-based approaches. But there is always belief. And this is the thing that scientists will often forget. There's always a belief element in science. Belief is subjective, not objective. And it's that belief that can lead us to some of the issues that we face when challenging paradigms. So, nitrogen fixation, what is the paradigm that we've been challenging for the last 10 years? That's one, and Ted Cocking, who I'll talk about in a bit, challenged for 40 years. The intracellular symbiotic colonization by bacterial nitrogen fixers is actually possible in the absence of root nodules. Okay? That nitrogen fixation can actually occur throughout a plant, and that amount of nitrogen that is produced is sufficient to maintain or enhance crop yields, and that in doing so you create a new plant organelle that is called a diazoplast, and that's Ted Cocking came up with that term, a nitrogen fixing organelle. So you probably all know about nitrogen fixation, bacterial nitrogen fixation with those bacteria and that are in the root system, that are certainly in the soil system. And we all know about rhizobia and root nodules. But of course, science has moved towards how do we take the root nodules that we know happens in all of our legumes, how do we create that in cereals? And that's been the big thing that has been, I suppose, dominated the paradigm, if you like, of nitrogen fixation. How can we use the latest scientific techniques of genetic manipulation to create cereals with root nodules? What if then people come along, a person in particular, Professor Ted Cocking, comes along and says, oh, that's not actually necessary. I found a bacteria that can do it all on its own without any root nodules. Then there's a whole industry out there of funders, a whole industry of scientific researchers whose interests suddenly become undermined. Do those scientists step back and say, well, that's fine? No, they try to undermine the competition. And that's happened on a big scale. This is the late Professor Ted Cocking, and he would have been delighted to know that I'm here talking to you today. He passed away at 92 last year. And those of you who of course know agriculture will also see that there's Norman Borlaug there on the right hand side. And Ted knew Norman well, and Norman Borlaug did encourage Ted to work on nitrogen fixation. How did Ted get into this? Ted Cocking is actually famous and got his Fellow of the Royal Society appointment because he worked out how you could break down cell walls and get things inside plant cells. Of course, we're talking about a fixed structure with cellulose. And so it's really a case of, he started saying, well, how can we work with plant cells in the same way as you can work with animal cells? And that was always his ambition. Then he realized that, well, could I use rhizobia bacteria and try to get rhizobia into plant cells and still get the plant growing up and fixing nitrogen? He never succeeded. For 20 years he did not succeed in achieving that, but he kept at it, he kept at it. So plant intracellular colonization, not intercellular, intracellular. Root nodules we know about with rhizobia. There are examples of bacteria fixing nitrogen in other crops or in other plants. So in Gunnera, the non-legume Gunnera, it's well known. Bacterial intracellularity, it's complicated, has been identified in roots and stems and leaves of a whole range of different plants. So you might ask, well, why is this so radical? We'll come to that in a bit. You've also seen bacteria in seed symbionts, endosymbionts. Nodules are not actually required for intracellular plant endophytic nitrogen fixation. But there is a community out there of scientists who have fought and ignored that. And we've come to the conclusion is that most plants can probably have bacterial associations and fix nitrogen. We just haven't looked. And the more we look, the more we keep finding. So how does that work? So I'll use the pointer thing. Quite simply, I'll talk about Glucocetabacter diazotrophicus in a bit. As a bacteria, what it does is it produces enzymes. The enzymes are released, punctures holes through the cell walls of the root, or will go in through if there are any abrasions or breaks, and it then forms here. It will get inside the cell by releasing these enzymes, create this vesicle around itself, an endosymbiont it becomes in that case, and that is what we will call the diazoplast. It's the bacteria when it's surrounded by the vesicle, and then that vesicle will reproduce and move throughout the whole of the plant. Now that's a very simple diagrammatic representation. Making that happen in the first place, in getting the bacteria to sit on the outside of the root, to then produce the enzymes, break down the cell wall, and then to colonize, that's the key to this whole technology that I'm talking about. Once it's in there, believe me, it does its stuff, and it's quite impressive that it does its stuff. That's what it looks like. So I need to say something about Glucocetabacter diazotrophicus GD. So it's first isolated by the Brazilians in sugarcane, and it is here in sugarcane in Australia. Absolutely no doubts whatsoever. It's found in sugarcane. The reason they found it was that they needed to increase their amount of alcohol that was produced, that they could produce from sugar, so they could replace and have a substitute for petrol, because at that time they had problems in access to oil. And so what they did, they started looking and realized that there were lots of sugarcane plantations out there that had never been treated with fertilizer, yet they still maintained their yields. So they started looking to see what was in there, and what they found was Glucocetabacter diazotrophicus. There are a number of key characteristics that I want to just run through, and I won't go into any detail. I can talk to you more about them. But first of all, this bacteria does not have a nitrate reductase enzyme, which means it does not recognize when other sources of nitrogen are present. Now that is brilliant, because it means you can even add nitrogen fertilizer and you get a synergistic effect. It will add on top, additive. It fixes between 60 and 80% of all the sugarcane needs in terms of nitrogen, and it delivers about 50% of everything that bacteria produces to the plant. GD has these enzymes which promote cell wall penetration. Not every isolate that we find of the bacteria has those enzymes. So we found that some work in association with others. It's not just about necessarily a single isolate, but the one we work with has got all of these characteristics. Massive, massive genetic flexibility through the genome and plasmids, and those enzymes talking about are in the plasmid. So not all of the bacterial isolates have those enzymes. It can respire under a range of oxygen concentrations. You can actually find this bacteria not only in the plant but in the insect gut of insects that feed on sugarcane. And in doing so, that has to survive in a wide range of oxygen concentrations. That is quite a rare characteristic. Has one of the highest respiratory rates of all aerobic bacteria. Why is that important? It's because you have to be able to protect the nitrogen, the enzymes that fix the nitrogen. They do not survive in an oxygen-rich environment. So that respiratory rate is key to that process. Interestingly, and I'll tell the story in a bit about a Professor Apposti, who is an Italian scientist. He works on mitochondria, and he's also looked at protomitochondria and where the derivatives of these protomitochondria come from. In his studies, he's identified Gluconacetobacter diazotrophicus as one of the potential candidate species as a protomitochondria because of this high respiratory rate. This makes this bacteria look increasingly interesting, doesn't it? And it's got a range of characteristics that help protect the nitrogen fixation process. And this is one of the big criticisms we get from those that are interested in rhizobia. Right, so let's see if this works. So one of the key things that we will be always being, and Ted Cocking was always being questioned about, was can bacteria actually survive once they're in the cell? So this is hopefully, there we have it. Don't get too excited. This is not going to be a big hit on Hollywood. That's a GD doing its thing in a tomato protoplast. So the walls have been stripped off, bacteria has colonized the plant cell protoplast, and it's in there doing its thing, reproducing. So that was the first thing to demonstrate, that you could get the bacteria, it could survive, it could reproduce, it could do its thing intracellularly. And this is work of Dr. Michele Palluccini in the University of Nottingham. So it'll carry on doing its thing for a little while. So one of the interesting things is almost this double standards you get with paradigms. Can you tell me which of those pairs of images, one is of GD colonizing a plant, a maize plant, the other is of a rhizobia colonizing a root nodule. See any difference? Really? Maybe which would you say is the rhizobia and which would you say is the GD? Any thoughts? Go on, be brave, somebody. So it's rhizobia on the right, GD on the left. Paper published, no problem whatsoever. Criticised, commented on, went through any number of referees who would not publish this because that image did not demonstrate bacteria in a plant cell. That one did.
That's in the roots, right?
That's in the roots, yep. So, does it fix nitrogen? Does it actually fix nitrogen? Does it do the job? So what we did is basically said, what are all the methods that are used for nitrogen fixation and measurement of nitrogen fixation by rhizobia? So we went through all of those, and there's a whole load of work that's been done by other people demonstrating nitrogen fixation using the same measures. So measurement of green foliage, chlorophyll content, measurement of percentage N, nitrogen in the biomass, labelled nitrogen studies, NIF minus mutant studies, so that you're switching off the nitrogen fixation gene capability and comparing it with plants colonised with those switched on and those switched off genes. Measurement of nitrogenase activity using the acetylene reduction assay, photosynthetic rate and water use efficiency, demonstration of field efficacy. We've done all that, others have done all that, and still we had problems in believing that we were actually fixing nitrogen. Colonisation. These are all the crops we've worked on with this particular species and isolate of GD, and we've managed to get colonization of bacteria in all of them. Every single one we've tried, we have managed to get colonization. Isn't that a bit scary? Quite an amazing list. So we make, rather grandly, the claim that we think we can get nitrogen fixation with this bacteria in every crop, because that's such a diverse range of crops. Wouldn't that be amazing? Of course, it's got to fix nitrogen to a level that matters. So here's a bit of the science on the nitrogen fixation. So this is one of the early bits, the chloroplasts up here, maize leaf, that Ted Cocking did. That's the bacteria, the black dots. And funnily enough, the one thing we find is that they seem to concentrate around chloroplasts in the leaves. Consistently find that. And I got some really exciting news literally a week before I came away to Perth, and I'll tell you about it in a bit. This is a work, so that's work by Ted Cocking. This work here was done by Karen Moore at University of Manchester. She's now moved from there, and the work is being continued at the National Physical Laboratory by Greg McMahon. This is using nanosims microscopy. What it basically is showing is anything that it has got yellow to red in there means it's picked up nitrogen from the air and it's fixed it. Okay, and you're seeing an isotope of nitrogen, and it's being fixed by the bacteria. These are the control bacteria in the sense that they're completely not a nitrogen fixer, and anything that's blue basically means there's no nitrogen being of the isotope being picked up and converted and fixed by the bacteria. Here, these are chloroplasts, and what do you see? Bright red, and that means that the nitrogen that has been taken from the air has been fixed by the bacteria, and it's ending up in the chloroplast. A paper comes out on this in the next few weeks. It's been a long old haul getting anybody to want to publish it, funnily enough. We've also done some work with Eric Murky at the University of Nottingham, and he's a photosynthetic physiologist, particularly on rice. He did some work on rice and started to show, and we've done more work on this since, that we get an increase in photosynthetic efficiency with this plants that are colonized with this bacteria. Why is that, you ask? We'll come back to it. And then we did some, University of Nottingham did some work on the nitrogen fixing genes, switching them on or off. And so the NIF minus genes, NIF means nitrogen fixation, the NIF minus means the genes have been switched off in the bacteria. There are 3 genes that are really important, and funnily enough, these are tomato plants. That's one of the genes switched off, another of the genes switched off, another of the genes switched off. So what's happened is the bacterial genes have been switched off in the bacteria have colonized the plant, and then we see what effects they have on the growth. That's the control plant, those are the GD genes have been switched off, and that's the colonized without the genes being switched off, with the genes still switched on, and the plant growth increases quite significantly. So that's demonstrating it is down to genes, it's not down to plant hormone effects, it is down to the nitrogen fixation genes. And so I'm just showing you a snapshot. There's more behind all of this, of course. This is where it starts getting a bit silly, because this is Rupert Frey, good friend and professor at the university, head of department at the moment, and this is a tomato plant that's not colonized. That's a tomato plant that is colonized with GD. That's the difference it made. I mean, just crazy. This is microtom. This is that's a control, 3 different concentrations of the bacteria, and that's the impact it has. This is Sophie Alexander, who's become a good friend, a UK organic wheat farmer. She did some of the first early work for us, and this is in her wheat. She's actually phoning me at the time that this photograph was taken to say, I don't believe what I'm seeing in my wheat field. And that was the difference between control and treated. No nitrogen fertilizer in sight. So how does that all pan out? How are we doing? How does that all pan out in terms of yield responses? So everybody in this audience, I'm sure, there's always an optimum N that you can apply, and after that yields will decline. So we expected this to happen. We thought, oh well, at the low N levels, the GD will compensate, there'll be nitrogen available, therefore we'll get an increased yield, and that will still level off at the optimum. The other possibility, and we did not think this was going to be likely when we first started, was that you get this parallel response. And we thought, well, that might happen if plant growth hormones come into play, as well as nitrogen fixation. You may get a change in the overall, if you like, physiological response of the plant to the bacteria. So this is the first trial we did, and we worked on grass. Turf grass. Why turf grass? It's hardly the big economic crop that we're going to be concerned about. My concern was quite simply that the bacteria could not move from the seed, which we were treating, colonize the plant, move through the whole of the plant within a season sufficient enough to impact on yield. So I thought, oh well, a perennial crop, grass. That'll be good. We can look at that in a number of different ways. And we were staggered to find I was completely and utterly wrong. NDVI, measure of the greenness. This is just a percentage increase versus the control, so we got nearly 10%, 12% there. Leaf nitrogen content went up, biomass went up. We were just really staggered because we didn't really expect to see this level of impact. Then over a number of years, we then conducted a whole range of different trials. Now this is when you're in the product development process, you're looking at optimization of formulation, optimization of colonization rates, the densities, the application rates you use. All of those things have to go in. 174 different trials in 10 different countries, and how many different crops was that? 7 different crops. So we got a lot of data, and this was work that was done. We paid as a company to have these trials conducted, but we did not take the measurements. These were independently assessed. So average maize field trial results, we got a 27% nitrogen saving on average, an average increase of 8%. But here you can see this parallel lines effectively in yield. This is not what we expected, but we consistently see now in every crop we work with. It's suggesting a change in the physiology of the plant because of the presence of the bacteria. USDA did some work for us on biomass and grain impact as far as as a forage crop was concerned, and they found we got a significant improvement in energy usage and digestibility of that maize. So we were then interested when we started getting these results in wheat. I know we have some wheat experts in the audience. So what we actually found was that we got a 61% nitrogen saving we could make and still maintain crop yields, and we basically again had this parallel. But what was really important is grain protein content increased on average by 4%. Now you think that might be great. The story gets even better because we go to the millers who make the flour from the grain, and we went to 2 of the UK big millers, and they basically looked at this, and one of the chief scientists of one of them said, my goodness, I've never seen a quality of flour as good as this in all his career. And so it's not just protein content, it's the ability to the flour to make the bread and the content of the bread. or the quality of the bread. Best results have always come with rice, which is curious. You may ask why, and this is because we find well, the key thing is colonization, getting the bacteria into the plant. With rice production, they tend to farmers soak the seed and it germinates, and if we soak the seed at the same time as we add the bacteria goes into the emerging root more efficiently. And then you get uptake, colonization is guaranteed. We get the most variation, as we understand it, by lack of colonization of a plant. Not once it's in there, it does its stuff. But getting it in there is the key, and that's the problem we face as a product development issue. How can you deliver this? Well, you can deliver it to vegetatively produced plants, quite simply. If you've got a cutting of cassava or of a yam or a potato, you can dip it and the bacteria goes in and it stays in the plant. We showed this with potato for 4 successive generations. We stopped after 4. 4 successive generations the bacteria stays within the plant. So that means for developing countries where you've got crops such as yams and cassava, you basically only have to treat it once and then the farmer can run with it and keep reproducing it vegetatively and propagating it that way. Seed crops, it's easier to apply this bacteria to a seed crop in a bucket than or in a cement mixer than it is at high-tech, ultra low volume application seed treatments. Keeping it alive on the seed long enough for the seed to germinate is one of the key things that we have to do with the product development. We've managed to do that. Just so you know, regulatory environment, really, really not an issue. And we're pleased to be able to say that it gets registered with organic organisations around the world, and there are no concerns at all. People have been eating GD for generations from sugarcane anyway, and all of the regulators. In the UK, you don't even have to, there are no regulations for it at all. Europe have got more that they're bringing in now. So we started out with an intracellular nitrogen fixation capability as a laboratory concept in 2011. We looked at the technical potential of that and formed Azotic Technologies in January 2012, and an initial formulation which, I hate to say it, was more of a finger in the air and let's just try this, that worked, and we did our first field trials in June 2013. I got lambasted as a scientist because we needed funding. We'd been given money by a Canadian entrepreneur to do one set of field trials, and that was it. That's all we had the money for. And so how do you get more money when essentially there are a lot of we tried to get grants from the government. The scientists who do not want to see a paradigm shift are those who are the experts who make the assessments as to whether you should get funding or not. We didn't get funding. So where do you go to get your money? So I went public, and I'm still suffering from that, because 1 year field trials data going public saying we've got something wonderful, and of course that's not what you would normally do. But it meant within 6 months we had £10 million worth of investment from a company in the Netherlands. That allowed us to go into a program of developing our laboratory, getting our scientists on board, and doing all of our field trials. That's taken it to where it is now. So proof of efficacy in Europe, North America, Southeast Asia, sales in 2019 in the US, followed by Canada 21, Europe this year, and Sumitomo Corporation have taken over the distribution for Europe. So it's come a long way. we formed the Sustainable Nitrogen Foundation in 2018 because we felt, since you can put seed in a bucket, put in the bacteria, stir it round and sow it, this technology is highly appropriate for African smallholder farmers, and we did not want to wait until the company got through its phases of going to all the major markets, then going to the smaller ones, and getting around eventually in 10, 15, 20 years' time to the smallholder farmers. We're shortcutting that process. I have written a book about this called Fixed on Nitrogen, scientist short story, which tells this story in more detail. All of the money goes to the foundation. If you were to buy a copy, you can get it on Amazon. So, has the nitrogen fixation paradigm shifted? In 2023, just a year ago, in May, I was invited to go to a conference organised by Bill and Melinda Gates Foundation. Because they had been looking at this, but they had been advised over many years that this was snake oil. So we got together all of those scientists in the US who work with endophytes, in the rest of Europe, in Africa, in Brazil, and we all got together, and they were very gracious at the end because they said this expert convening concluded the endophytes are more reality than myth. Well, thank you very much. We were very pleased with that endorsement. So what if every green plant is capable of an intracellular symbiotic relationship with a nitrogen-fixing bacteria? What if we could get that into every crop plant and fix nitrogen without or with reduced need for nitrogen fertilizers? Why shouldn't we be able to, given what we've heard today? I hope you agree. And what about modern crop plant breeding techniques? We have some evidence that suggests that historically older cultivars are easier for the bacteria to colonize than the modern ones. And we know from work in Mauritius with sugarcane that they'd bred out the ability for the bacteria to sit in their elite cultivars. We're now working with them to get the bacteria back in again. So, I went to Tim Minchin on Sunday night. I don't know whether you like him or hate him. I think he's absolutely brilliant. But I couldn't resist this, to finish on this quote, to put into the world valuable ideas. Look for stories that are worth telling, lessons that need teaching, and tell them and teach them, and stay passionate. I am passionate, and I think the last line is even better. I'll see you in the pub. Thank you very much.
Thank you, David. Thank you for that interesting and wonderful story. So we'll take some questions because we have time. Who wants to ask? Jan Pan.
Hi.
That's very interesting talk. I have a couple of questions. So out of so many nitrogen fixing strains, why did you particularly focus on one strain called GD?
Mm-hmm. Yep. Okay.
And then the second question is, I have mentioned that those strains can actually pass, if you cut something and pass to the next
Generation,
plant, the generation. So my question is, so for example, when you grow this plant for one season, do you think there will be any remaining for the next season or effect on the following crops?
Okay, right. So Ted Cocking worked for, must have been about 55 years on nitrogen fixation, or seeking nitrogen fixation through intracellular colonization that was not using root nodules. He screened any number of bacteria, and he was very interested, of course, when the discovery in 1988 by the Brazilian scientists that this bacteria GD. So he pulled as many of these isolates as can be found by because a lot of scientists started looking on that basis, and he started screening them for what he thought was the characteristics. And what he was looking for was the presence of the enzymes to break down the cell wall, and the ability, obviously, to fix nitrogen ones inside the plant. And he went through a whole range of different strains, and he came across just one. Talk about serendipity in science. One strain that fitted all of that criteria. And so he started working on that. And so that's why, yeah, I mean, it's just one of those things. He tried working with the other azospirillums and all the rest of it, and none of those would fit the bill. So it's finding that one that did. And I still to this day believe there are many out there. In fact, all of our research that we do in Africa is screening for local isolates of GD and other bacteria in sugarcane, and seeing which ones of those are most effective. So that's the first question. Is that good enough answer? The second question is really, really interesting, because what would be great is if you could put the bacteria, not from a commercial point of view, and I have to say I am an entrepreneur, so I like to see that there's business and commerce involved, but from as far as the world's concerned, wouldn't it be great if the bacteria could go into a wheat plant in one generation, get carried over in the seed into the next generation and grow up, and so on and so forth. We did work with oilseed rape and found that it seems to be, and you maybe know more about this than I do, but essentially it seemed to be a random process as to whether the bacteria got into the seed or not. So in oilseed rape, we found from colonized plants, one generation, next generation, only 18% of those plants had the bacteria present, next generation, none. But in potato, in cut plants and vegetative growth, where you're doing it through, then it's possible to pass it on to the next generation.
I think the other thing regarding that question, the GD is not just specific to one plant. You showed a diverse plant, so that's another. There is one called azolla, which is growing in the rice field. That may be too specific to that. It works wonderful. Other questions? There's one from the
Excellent talk, David. Thanks.
You're welcome.
Have you done any work with metalobacteria, and what's the parallel to them to GD?
And the answer is no. We haven't done anything, and no, I'm not qualified to say.
Yes.
What would be the main food for these bacteria into the protoplast?
So the main.
What is the resource? The food for the bacteria.
Yeah. So sucrose.
Sucrose.
So I mean that all fits simply because you find them in sugarcane, and so we've always used sucrose as the energy source, and that's why we think we also get the bacteria sitting around the chloroplast, because that's where the highest concentration of sugar is going to be, and so that all ties up. But the thing I was going to tell you that I didn't, but that just before I came away, a colleague in Rutgers University in the US emailed me to say, we think we've found the bacteria inside the chloroplast. Wouldn't that be incredible?
Yeah.
Hi, thanks for the excellent talk. I'm interested in protein quality. So you were mentioning that there was bacteria actually changing the building quality or the flower quality.
Yep.
So just wondering, did you do any further measurements? So we're doing a lot of like mass spectrometry, for example, to kind of see if composition of proteins, like storage proteins, actually changed.
No, but we're looking for people who can. So no, I'm being flippant.
Just interested if you actually saw changes in storage proteins.
I mean, there's just only so much we've been able to do. We've done some studies to look at essential amino acids and found that we get lysine, for instance, is increased massively with the presence of the bacteria. We don't know why, but lysine is increased. There are a couple that are lower, but for the most part it's the same, but a few key essential amino acids are increased, and we've got no idea why. But we're at the beginning of that story.
One of the things with very good with the nitrogen application is that is applied either at seeding, and then it has to be taken up and then translocated. By the time the grain filling and other things comes, there's an efficiency loss. Whereas if this is within the leaf, let's say flag leaf of a wheat plant, that's when the grain is started filling down. There could be much more effectiveness of that nitrogen in relation to protein synthesis and then laying out. But you mentioned about the dose strength and the bread volume and all that. That's interesting. Other questions? There's one here. Yeah, please.
Yeah, first of all, thank you very much for the talk. I have two questions. First one is that, is there a research that try to explain why this microbe is very general, that they can infect so many plants?
Yeah.
Because as so microbiologists, they believe in specificity.
Yeah.
So the generalist concept is pretty much not popular. And then the second one is that, from commercial point of view, if you put this GD and the rhizobia side by side, then from your presentation, it seems like GD is winning on everything. So is there still some advantages that rhizobia still have over?
Okay, I'll answer your second question first. The rhizobia, there is synergy between the two. So rhizobia can work in a legume, and you put GD into the legume, there's no competition that we can detect. You actually get a synergistic effect. Because GD does not recognize the presence of nitrogen, it basically just keeps doing its thing wherever it is. The generality of GD is a surprise, and it's really a question of the issue is always about getting it inside the cells of the plant. Once it's in there, then there doesn't seem to be an issue. We do get variation between varieties of crops. We do get variations under different conditions. So there are factors that are affecting it. We just don't have a good understanding of it yet, but under laboratory conditions we can certainly get the bacteria into any crop. Once it gets into the field, it gets more variable.
Thank you very much for a very interesting talk, and I have personal experience of working in fields where it's hard to change the thought process. But what I was going to say was that it seems to me that there's an increasing number of organizations, companies and others, that are looking at adding bacteria and other organisms to the soil to improve plant growth, particularly crop plant growth.
Yep.
And that from a business perspective, you might have a lot of opportunity with some of the larger companies. I know that they do seed coating, but obviously that's not working, or it seems not to work too well with a GD type application.
No, that's right, yeah.
But it looks like there's probably plenty of opportunity out there
Yeah.
to have a look at other companies. And yeah, it's interesting. I find it personally very interesting because I was talking to people last week who were interested in cytoplasmic cell sterility and organelle transformation. So
okay.
it's sort of been a very nice talk for me from that perspective.
Oh, well, I'm glad to hear that. And just to comment on your point, the nitrogen fertilizer market, or if you like, the nitrogen market, is massive, absolutely massive globally. And there is no way that Azotic Technologies is going to dominate the world as having the only products that are ever going to work in this space. There are multiple solutions to the same issue. We have one of them. We have one of them that works very well, but has issues. So once we've got it in the plant, no problem. Others, broad spectrum, in soil, microbes, they have issues with if it's too dry, too wet, too hot, too cold. We don't have that issue once we've got it inside the plant. So it's always going to be a mix, and we find that there is synergy as well. So it'll always be a combination of can we use microbes more effectively in the soil and those that are endophytes as well, and get those in combination, and then we've got the best of all worlds.
But the biostimulant is one of the, I'll come back to you. Biostimulant is one of the growing markets,
Yes,
even in your
absolutely.
own country.
Massive,
I know,
yeah.
so it's massive,
Yeah.
especially its application in developing countries and small farm holders. One of the things which happens now, seed priming is primarily used. I've published many papers and reviews on seed priming. Whether you have micronutrients supplied, and many of the smallholder farmers can handle that seed priming, whether it is rice, wheat, or other crops. It's a resilient technology because it's smallholder. Luis.
So what other factors influence the success of colonisation?
First of all, you can try to get it into the seed. So when the seed takes up water, then the bacteria go in as well. What we've found there, though, you have to be very careful on the dose, because you can, if you imagine it, it's going to use sucrose as an energy source. So those early stages of a seed growing, it needs all the energy and everything, and you're actually adding a burden. So you have to be very careful with that. So getting the bacteria to stay alive on the outside of the seed, when the root emerges, then the bacteria starts to colonize. It goes at a steady rate, and for the most part it's not an issue there. But that balance between affecting germination and affecting and getting sufficient bacteria in. What Ted Cocking actually demonstrated, and the grass trials I showed, we only added effectively 50 bacteria per seed. Incredibly low numbers. We now add millions, because we're trying to compensate for a whole range of different conditions and all the rest of it. But we started out from a laboratory treatment. Now I will emphasise that, laboratory treatment, 50 bacteria per seed.
That's pretty small compared with the rhizobia.
Yeah.
Any other questions? Yeah. Dana.
Thank you. It was a really interesting talk. So you registered the charity in 2018 with a very noble mission to improve smallholder farmers in Africa. Can you talk about what progress you've made
Yes.
over the last 5 or 6 years?
Yeah, delighted to do so. Yes. So setting up a charity is a bit like setting up a new company, and it takes time for everything to get into place and to get the resources you need. So we actually have had, we won an award from the UNDP, which gave us funds to set up a project in Gabon. We then have got funds to work in Rwanda, and we're now into field trials in Rwanda, where we've isolated the bacteria from sugarcane. So it's indigenous GD, and we've gone through a process of screening to find which are the best ones for those particular crops that they're interested in. And we're now taking that forward to extensive field trials this next year. So that's gone really well, and that will continue for the next three years. And what we hope to be able to do is to produce GD in country using indigenous strains to cut down on import costs. And you might think this is counter to my company, and I'm a shareholder but a non-voting shareholder now, so that I could set up the foundation. So the reality is these are small markets that really are not of concern. So we're help them produce and manufacture within country their own indigenous strains. That's in Rwanda. We've just got a new project in Senegal to do the same. And we're working with a whole range of big donor agencies to take this further and expand it.
Yeah, I think you are on the target, because if you look at the global fertilizer consuming continents, Africa is still one of the least. You're talking about average less than 10-15 kilograms of nitrogen. So it's the right target. And again, I think this kind of technologies have got immediate application in smallholder farmers, where they can do all this kind of treatment. When it comes to bigger situations like Australia, it is harder for this treatment, but it's worth. My question is that have you got this material already in Australia where we can get a handle? Because a lot of our students, including some of my students with the Jain and others, are looking at endophytes. We know the study I said, one which I published on Pseudomonas.
That's right. Yeah.
And how wonderful. I didn't believe about it, but these things can go and do some miracles in terms of changing the composition, in terms of resilience, etc. So Can we get that, and then we will just put on to one of our students who is already looking phytate in wheat, for example. I mentioned to you.
Yeah, no, that's right. So I'll have to contact Exotic on your behalf, but I know Australia is obviously one of the, I hate to use commercial terms, a target country, so I don't know how far.
It is the quarantine is the why worry, because otherwise I can just write a letter and get it, but the quarantine is the biggest
but
issue.
that doesn't tend to be a problem simply because down your east coast you obviously have sugarcane, and the GD will be in there. Well, we know that's the case because Australian scientists have isolated it from there. So.
But we want to get your product so that we can then do that and study it, because it's very
well, and I'm sure if you contact them direct, they will send you samples. I'm sure that's possible.
We'll do that. Any other last questions? Otherwise, I'll wrap up. Thank you very much. Again, please join me in thanking David.